Hello, hello, hello. It's Wednesday, so you know what it is. It's Life Sciences with Looney and Cheryl.
Good evening, everybody. We
Still trying to recover.
We had a voice from above. Just about.
You're such a shock. But anyway, guys, welcome grade 10s. What are we doing today, Cheryl?
As I said to you, we're looking at the leaf.
Such complex things today. Okay, mindset is you heard it. We are going through the leaf. Just in case you guys don't know by now, we are giving away Casio calculators every show. So for this show, all you need to do is download the show notes. I have posted the link, and then all you need to do is complete the test yourself section. But that is, you complete the section on the entry form. So all the questions are on the entry form, and then you just complete that. Then you'll stand a chance to win this awesome Casio calculator. Simple as that, guys. And all the questions are based on what you guys are doing in life sciences. So it's not like you need to come up with these magical answers. No, it's stuff you know. So make sure you do enter the Test Yourself competition, probably brought to you by Casio. What else do you need to know? Hit us up on Facebook and on Twitter at Learn Extra. Then you can download the show notes, the videos, and the schedules on learn.mindset.co.za.
Now, we're also taking a collection of money in today because Looney's pants seem to have splits in and our manager's shoes are broken. So any clothing donations are quite welcome at this side. Welcome, grade 10s. I'm going to do a shout out for the first time ever. I got ambushed at the end of school today by two ladies, Tegan and Ashley. All right, they said, ma'am, are you on TV today? I said, yes. They said, shout out. So Tegan and Ashley, shout out. But I'm going to do own personal shout out for the first time ever to my little pinky tink at home, Callie. All right, it's her birthday tomorrow. Happy birthday, my sweetie. I hope she's so excited. One more sleep until she's 7. And I'm sure she's going to have a wonderful day. Okay, enough of that personal stuff. Right, we're going to start with the leaf. Now, if you're wondering where does it fit into everything that we've looked at, let's start at the beginning. All right, we looked at the cell, which is the basic unit for all living organisms. If we put cells that are similar in structure and similar in function, you get a tissue. All right, and as you know, we've looked at animal tissues and we've looked at plant tissues. Now, if we put all those tissues together, all right, we are going to get organs and we are going to get systems. But in plants, we're going to look at the organs. Specifically, we're going to look at the leaf, all right, today. Then we're going to look at the stem, all right, and we will look at the stem and the root together, all right, and the leaf we're going to look at a bit later how it's involved in the transport as well. So what we're looking at, we've done the tissue. Now let's see, we've done all the xylem, we've done the phloem, parenchyma, sclerenchyma, all these funny things, all right? But what do? They make up the actual plants. So let's have a look, all right, to see where, all right, the tissue is in the plants. We're going to be focusing on the leaf, we're going to be looking at the leaf, and we're going to be looking at the leaf particularly, all right, according to its major functions, which we are going to get onto. First of all, let's have a look at our challenge question. I think I've got the right order today, hey Looney?
Yes.
Because usually I forget about the challenge question. Right, guys, it's as simple. I always like just do the challenge question, just to put a multiple choice question in, just to let the brain juices flow a bit. Okay, over here you're going to become quite familiar with this diagram. It is, all right, a transverse section through a dorsiventral leaf, and you're going, huh? Right, we'll explain that just now. The diagram below is a cross section of a leaf where the arrows represent the net movement, all right? Be careful when you read your questions. Net movement of two substances. These substances, X and Y, are in order and there. Water or oxygen, water and carbon dioxide and oxygen, and oxygen and carbon dioxide. Right, look very carefully to see what you think your answer is going to be. Okay, guys, this is a very important, there we go. Let's get it nice, and this is the internal structure, right, of when we're looking at dicotyledonous plants. What do we mean about a dicotyledonous plant? Within the seed, those of you next time, if you have beans, all right, you can play around with your food. A bean seed, if you open it up, it splits into two. Di means to two, all right? So if we open up, we split it into two. And because it has two, as opposed to, the monocots 1, it's going to have, the roots are going to be different. Fibrous roots as opposed to taproot. The stems that we're going to look at next week, all right, or the week after, they're going to look different. The flowers also have different structures, right, and so does the leaf. When we look at the leaf, we call it dorsiventral, all right? What do I mean by a dorsiventral leaf? All right, dorsi means top. Let me do it over here. Dorsi means top, and ventral, all right, means bottom. If you have a look at the leaf, you're going to notice this is the top of the leaf here. There we have the top, and here we have the bottom. And if we have a look at the structure of the leaf from the top, that would then be the middle, all right, to the bottom, we will notice that the top is slightly different to the bottom. Dorsiventral, all right, meaning that there is a distinctive top and a distinctive bottom. So there's a top of the leaf and the bottom of the leaf. Next time when you go out walking, have a look. All right, leaves are all around you. Okay, have a look at it. See if you can see, all right, the top. Now, what have we done? We've looked at plant tissues, all right? We've spoken about what do we find on the outside? Our epidermis. What do I need, all right? What do I need to transport water? Xylem. What do I need to transport phloem? All right, food, phloem. So we've looked at all these tissues. Now we actually need to have a look to see where do they actually fit into the organs that we are going to look at. All right? So if you have a look at the organs, not the organs, let's have a look at the tissues, and then we will go specifically to see how is the leaf adapted to its particular functions. All right, there we go. All right, our first one is our epidermis tissue. If you have a look on the top here, all right, that is a cuticle. Now the cuticle, next time you have a look at the leaf, feel the top. You usually find that it's very smooth. We usually use the word waxy. All right, it's a waxy layer. It's a layer. It's not a cell layer. It's just a waxy layer on top, and it's there, all right, to ensure that the leaf doesn't dry out, etc. There too for protection, but it's not a cell layer. Now, underneath here we have our epidermis. Now, our leaf, we're going to look at, as I said just now, the main function of the leaf is to photosynthesize. So the cells within the leaf need to be adapted for photosynthesis. Now, when we looked at parenchyma, right, we said that some parenchyma has got a specific function. All right, and here, when we look at the palisade, these are all parenchyma. What's my parenchyma? They're thin-walled, all right? They've got intercellular air spaces in between them. Now, because these, all right, are going to photosynthesize, they are chlorenchyma, all right? Chlorenchyma. So they are parenchyma. See all the green, all right? All the green, right? They are parenchyma that are specifically adapted for photosynthesis. Okay, you're going to look at photosynthesis much greater detail when you are in grade 11. One of the things you need for photosynthesis, right, is water. So if you have a look here, all right, what are you going to notice? Okay, you need to have water and phloem getting up to the top. So you've got, there we go, you've got your two vascular tissues. All right, so we've got one. We're going to have our xylem bringing water, and all this green is going to make nice food that the phloem is going to take away. Okay, if you noticed, again, just all the tissues that we've looked at. Now we take those tissues and we put them in specific orders depending what is necessary for that particular organ. Okay. What you will also notice, there's a lot, all right, there's a lot of air spaces, and when we look, and we're going to look at them just now, we're going to see, when we look closely on the lower surface, that the epidermis on the lower surface has got very special epidermal cells. All right, we're going to find the normal epidermal cells, but we're also going to find those special epidermal cells, which are called, all right, your guard cells and your stomata. Sorry. There we go. What are we going to see at the bottom here? We're going to see these kidney bean shaped cells. And most importantly, they've got a hole in the middle called the stoma. All right, and you need to start thinking to yourself, a hole. All right, it's a gap. So obviously, things must move into the leaf and things must then move out. So if we have a look again at our cell layers, we have an epidermis, a lower and an upper epidermis. We have specialized parenchyma cells in the middle of the leaf. That's why we call them mesophyll. Meso, all right, middle, in the middle of the leaf. And then what do we have? We need to have our transport tissue, my xylem and my phloem, bringing the water up, taking the food down, and especially on my lower epidermis, I need to have specialized epidermal cells that are able to open and close so that water, oxygen, carbon dioxide, whatever, all right, might be able to move through. So when we're looking specifically at the leaf, all right, we need to know, and you know I'm very structure versus function. How is the leaf, all right, how are the tissues of the leaf adapted so that they can perform their function? I've started to mention the first one. Okay, here we go. There's your diagram of the leaf. Let me go to the question here. How is the leaf adapted for photosynthesis? All right, the leaf's main function is to photosynthesize. And even though we don't need to know the whole process of photosynthesis, we do need to know, all right, how certain elements that are necessary that the leaf enables this process to occur, all right, at a much, well, let's say, more beneficially. Let's have a look here. If we have a look at the leaf, all right, we're going to start off at the top. At the top, we have the cuticle and we have the epidermis. They are the very top layers of the leaf. Now, when we photosynthesize, what we need to allow light, right? Photosynthesis is a process that light, all right, is necessary. So now, what do I need for photosynthesis? I need these little chloroplasts, okay? Because the sunlight is going to trigger them. But the sunlight has to get to those chloroplasts. So it needs to be able to go through those cell layers, okay? So my cuticle and my epidermis, to be able for sunlight to go through, all right, they are transparent. Sorry, I've run out of space there. They are transparent. What do? They allow the sun, all right, to come right through to my palisade tissues. Okay, now my palisade tissues are where the action happens here for photosynthesis. Okay, when we talk about palisade, some of you might realize like palisade fencing. Palisade fencing is fencing that has these almost like the long poles here. If we have a look at the palisade, I'm going to draw them in here. All right, they are elongated. You will also notice that they are right directly underneath the upper epidermis. We don't really find them at the bottom. Why? Because sunlight is not usually doesn't come up from the bottom, unless it obviously refracted, but we're not going to get there. So what do we find? Right underneath direct sunlight, we're going to find our first layer of palisade. Now, if you have a look, when they are elongated like that, we say they have an increased surface area. All right. So what does that mean? Very simply, it's got a greater amount of area that the sunlight can trigger. So more area exposed to the sun. So the more area exposed, the more sunlight, all right, it can absorb. And of course, What are the organelles that are responsible for photosynthesis? Right, chloroplasts. Do you think it's going to have one little chloroplast per cell? No, all right? Its function is photosynthesis. So what do? I pack my cells full, all right, of chloroplasts. I pack them, all right? I pack them. And the more chloroplasts I have, the more chlorophyll I have, the more photosynthesis I'm going to be able to, all right? More photosynthesis can occur. One of the other things that I need, photosynthesis, I need water. All right, what do I need? I need water. How do I get that water? Xylem. Right, xylem. My xylem is going to transport my water. Right next to xylem is phloem. When I'm photosynthesize, what do? I make starch. Right, I make food. And that food doesn't stay there, all right? What it needs to do is the food needs to go down to the roots to be stored, right? So what do we have? We have phloem, all right, to take the food down. All right, and now if you'll have a look at all these little, all right, cells over here, that's the spongy mesophyll. If you notice, they're full of air spaces. What do we need for photosynthesis to occur? We need gases. We need carbon dioxide, all right, and we're going to release oxygen. So I need to make sure that all these spaces here are filled with all of these gases. Now I need these gases. Where do I get them? Aha! On my lower surface, what do I find? I find the stomata, all right? And what do the stomata do? They enable the gases to come through. So everything that I need, every single thing, all the raw materials I need for photosynthesis and everything that I'm going to release, all right? The leaf is equipped, adapted for all of those. We're taking a break because I see my new pieces coming for Dean.
Yes, we're taking a break. Quick shout out to Sia and Dilibanya and Gogetsu. Thank you so much, Mindsetters, for tuning in. Let's take a very quick break and we'll see you straight after this. Welcome back, Mindsetters, from that break. Cheryl, why are you daydreaming now?
No, I'm trying to think why my ear is like made so funnily that this thing never. Last year I had smartboard issues, and now it seems the smartboard's fine. Now it looks like I have ear issues. Yeah.
I was wondering why you're looking into the distance.
To myself, oh, I can hear nicely all of a sudden. Yeah, I wasn't daydreaming. I was just waiting for the proper introduction.
Okay.
All right, guys, we're carrying on with how the leaf, all right, is adapted. Oh, this funny thing. I think I got a funny shaped head as well. All right, how the leaf is adapted to gaseous exchange. That's the second, all right, the second function. The first one was photosynthesis. The second one is gaseous exchange. And I'm sure by having a look, we're using the same diagram again. All right, there we go. Sneak little preview. Okay, gaseous exchange, right, is going to occur here at the stomata. Now, if we have a look, I don't know if your teacher did it with you when you did plant tissue, or you're going to do it now, all right? But the stomata are specialized epidermal cells. When you did the epidermis, you might have done that. It's probably a regular feature. Those of you might have looked at an onion cell, all right, underneath the microscope, and you would have noticed something like that. But sometimes on the outside, the plant needs special adaptations. And I think what you would have done, I don't know if you, as I said, I don't know if you did it, you would have known that in the root, we needed a root hair, all right, a specialized epidermal cell to absorb water. And in the leaf, we need to have a specialized epidermal cell that needs to be able to open and close. So if you have a look at the stomata, all right, they are specialized epidermal cells. Here are my normal, all right, and there are my specialized. And if I look at them, all right, I'm going to see that one, they are a different shape. We say they are kidney bean shaped or bean shaped. You will also notice that the inner wall here is thicker than the outer wall. And if you have a look carefully, you're going to see that there are going to be chloroplasts inside there. They can actually photosynthesize. And it's not to make food, all right? And there's a question later in one of the exam type questions that are asked, all right? Is how does the stomata open and close? And it opens and closes, right, by these chloroplasts in the guard cell. Okay, I just want you to, I'm going to give you a little hint before we carry on with the questions. Chloroplasts photosynthesize. They make glucose. All right. Look at the cells on the outside. They don't make glucose. So we have a high water potential. Is osmosis coming back to you? All right. Versus a low water potential. Where's the water going to move to? All right. So it's going to, the movement of osmosis, right, that's going to be how the stomata is going to open and close. But when I get to that question there, I'm going to explain it to you. All right, much more clear. I don't want to give the question away. All right, so what you need, gases. Gases, the leaf can't absorb them through the epidermis, all right? Doesn't have that kind of function. The epidermis is there to protect. So we need to have specialized cells. So how is it adapted? All right, it has specialized cells, the stomata, right, which allow for gaseous exchange. And when we talk about gaseous exchange, you're going to see that it's not only carbon dioxide and oxygen, all right? Water vapor, which is a gas, water vapor also goes out, and that is also, all right, gaseous exchange also goes out, and we're going to look at that later, all right, in the series, because that is transpiration. Very often when we look at the leaf, we're going to link it to the process of transpiration, the transport of water in the plant. So there, and what else? How else is it nice and easy for gaseous exchange? What do we have a lot of, right? We have a lot of intercellular air spaces or air chambers that can store the gases, right? So the gases, much easier for them to move in and out. Okay, so number one, how was it adapted to photosynthesis? Number two, how is it adapted to gaseous exchange? And then the third one, right, that we are going to look at is how the leaf is adapted for transport of water and food. Right, at the beginning of the leaf, when we looked at the structure, we also said, and we'll link to photosynthesis as well, right? Photosynthesis needs water. So what do we have? We have xylem. All right, and how is xylem adapted to transport water? Remember, it is hollow, it is thin, right? It is strengthened with lignin. Okay, so we have, I always remember the straw, all right? When you think of xylem, you must always think of the straw, and that's what it does. From the bottom, and you're going to look at how that water transports up later. From the bottom, from the roots, the xylem is going to transport the water up to the leaf. So how is adapted for transport? The xylem, which is adapted to be able to transport water to its fullest. Sorry, what does photosynthesis do? Photosynthesis makes, I'm just going to put here food, right? We can say starch, right? Glucose initially stores its starch. But what do we want? We can't keep the food, all right, in the leaf. We want it to go down to store. We want it to go down. We want water to come up, and we want we want the food to be able to go down. And how do we do that? We have got the phloem, right? Go back to your plant tissue. Phloem is not the same. Xylem, long, thin, right? My phloem is not quite the same. It has got these sieve plates in it. It's got cytoplasm, and you can imagine treacle. Imagine the food, all right? The food going down here. When we look at transport, you go, oh, I've got an itchy nose. I think somebody's going to fight with me today. You keep laughing, all right? The food travels through that, and it trickles down. I'm sure you've all taken some syrup or some honey, right? And you've just twirled it around. It doesn't go like a glob. It slowly trickles, and that's basically what the food does, right? That is called translocation, but we're going to deal with that later. Are you sitting there just...
I'm
Are you listening?
To you. Yes. Because you always complain that I'm not concentrating.
No, last week I needed you to concentrate. I was like, come, Looney, be on the ball here. All right, let's have a look at some questions. Okay, you can see I'm fiddling with my earpiece again. There we go. Now I feel like an alien. Okay, we're going to deal with some questions, exam type questions. Okay, again, bringing in, very often when they do the leaf, okay, remember, it's plant tissue. Think about your plant tissues. First of all, be able to describe. Notice that it's a leaf, right? Because you're going to be doing the root, you're going to be doing the stem, and you're going to be doing the leaf. And you might get all three diagrams in one question. You need to be able to know this is a stem, this is a root, and this is a leaf. And don't say, yeah, they all look the same. Trust me, sometimes when you test, that all of a sudden everything looks like everything else. Okay. Let's have a look at our first question. Our first question, all right, examine the photograph below showing part of the epidermis, all right, of a Lindstrom leaf. Doesn't matter too much about the species name, but we know that it is the epidermis of a leaf. If I start to have a look, I will notice some cells look like this, and other cells I can see are slightly different. Okay, so let's have a look at the question. The cells labeled A, there we go, are modified for a particular function. State their function and explain how they differ from the rest of the cells forming the epidermal tissue of the leaf. You will see this question is similar again to one later on as well. It can be quite a common question. All right, so for 3 marks. For 1 mark, all right, for 1 mark, let's see. First of all, it says state their function. Don't get so lost, right, that all of you can think about, all right, you need to start off. Number 1, what is their function? And their function is gaseous exchange. All right, there we have. First of all, there we go. Then it has a second part of the question. Explain how they differ from the rest of the cells forming the epidermis. Explain, right? Explain. So what did we say, how they're different? Let's have a look at the diagram, all right? There are a few differences that we can mention, right? 3 marks, they're probably only looking for 2, but there are a few. We can list all of them because they don't ask us to be specific here. So if I was looking, what were the differences? Okay, number 1. Now you must be specific. Don't say it is bean shaped, it is not bean shaped. What is an it? All right? So we say the guard cells are bean shaped, kidney shaped. All right? You can use whatever. Normal. You don't have to say on normal shape, the normal on ot. Okay, next one. What was the difference? Okay, guard cells. If you want to put it in a table here, you're also more than welcome. Guard cells have chloroplasts. Epidermal, not. All right, another one. If we have a look, remember when we looked at the cell walls, all right, we noticed when we drew our guard cell, okay, that the inner membrane was thicker than the outer. So let's go back here. Another one you could say, okay, is in the guard cell. I'm just going to put GC there. The guard cell, all right, has a thick inner and thin outer membrane. All right. Whereas my epidermal, we can just say here is evenly thickened. There we go. Nice. Right. Remember to read the whole question. What is the function? And then explain. Explain. That is important. All right. Explain. Discuss. Describe. Explain. Don't tell me it is bean shaped. All right. It has got thick. It. Please be specific. The guard cells are bean shaped, whereas the epidermal are not. The guard cells have chloroplasts. The epidermal doesn't. Right? If it would be easier for you, all right, sometimes when you asked for the differences, some people are very particular. You could have put them into a table. All right? The epidermal and the guard cells over there. Now I've put in a skill, all right, for the next one. Now when it comes to, I'm not too sure if you do, but I know if I'm looking through most textbooks, all right, you do need to be able to have certain, all right, mathematical skills, calculation skills, if I can put that, okay? Now, let's have a look at the question. It asks you to calculate the actual diameter of the epidermal cell of the lines labeled X and Y, show all working out. Some of you, and I'm one of those, tend to shut down a bit when it comes to numbers. I'm not a number person at all. So when it comes to calculations, they tend to scare the living daylights out of me. Right, but if you have a look, you're asked to find out the actual diameter. Now, you should all know by now, in life science, all right, we call it the dam triangle. All right, the dam triangle. This is drawing size. This is actual size. And M stands for magnification. All right. And what they want you need to have two of those things. You need to have D, A, or M, all right, to work out this, all right, to work out this calculation. If you only have one, then it goes to the actual size calculation where you need to have, remember you have one of those scale lines, but we don't have that. Do we have two? Yes, we do, all right. I've asked you to measure from X to Y. So you need to take, and it's always better to measure in millimeters. It is far more accurate, okay? So you need to take your ruler. You need to be very accurate. You put it right there at the end of the thick lines, and you measure. When you measure from there to there, what are you measuring? You are measuring your drawing size. Okay, now when we start off, what are we looking for? We are looking for A. Now I always go back to my triangle because I told you numbers for me are, right, what does a triangle mean? That means divide, that means time. So A equals D, what does this mean? Over M, it means divide. What is my drawing? I take my drawing size and I'm just going to make up one. So you're going to measure. And say I measure that it is 20 millimeters. All right, I always write my units of measurement in. If I don't, I might lose marks. What is my magnification? It tells me. There we go. Here, I could work out the drawing size. Here, it gives me the magnification. All right, which was, I've got a short memory. Okay, 380. Here we go. 380, which gives me an answer. Now I'm going to use the calculator feature. I've never used it before. So if everything goes, there we go. All right, calculator, one of these that you can win. Okay, so I say 20 divided by 300 and, was it 380 or 360? 380. Oh, I'm telling you. Let me just see where it was. I've got going Alzheimer's here. 380 equals, 1 over 19. Remember, you need to press 0,05. All right, 0,05. What is it? Yes, millimeters, because their actual is a distance. So I gave you the 2. You could then work out the third one. All right. Luckily, it's only worth 2 marks. So those of you who are a bit frightened of numbers, right, don't run screaming just yet. Okay, now we have a look at the next question. Study the diagram of the cross-section through a die-cut leaf and answer the questions that follow. As you can see, being able to look at all the tissues we've just looked at. All right, give the main function of this plant organ. It's the leaf, its main function. These are all nice and simple questions, right? Photosynthesis. All right, provide labels for parts numbered. Now, again, this is just simple studying, right? Being able, knowing your leaf, drawing, and you need to remember, life science is a visual subject. It is pictures, right? You need to be able to know your labels. This is such a nice, easy question if you are able to label your diagrams. Okay, no interpretation needed here, especially of those of you who battle. Right, provide labels for parts labeled 1 and 2. So let's have a look. All right, 1 is my cuticle. I'm going to write it on here. Okay, cuticle. No, it's not. I apologize. Number 2 is the cuticle. Oh, and I know there's an eraser somewhere. Oh, my skills with the smartboard are just so awesome today. Don't tell John that. All right. Number 2 is the cuticle. Watch it all go blank just now. Number 1, it's not epidermis. All right. Please be specific. Upper epidermis. Some teachers might use the word abaxial and adaxial, bottom and top. Abaxial, all right, on the top, adaxial at the bottom. Okay, so 1 and 2. Upper epidermis, cuticle. Number 9 and 10. What is number 9? Let's find number 9 first. Right, over here. Okay, number 9. What will it be? Right, number 9 is a nucleus. Guys, hello. All right, nice and all right, labeled already for you. Number 10 and number 12. Number 10, you can use the word air chamber or intercellular airspace. Air chamber is better. And number 12, it's showing you the whole structure. So it doesn't want guard cell, doesn't want stoma, all right? It's showing you all of that. So stomata. Remember, this is the guard cell. The whole is the stoma. Together they form the stomata. Right, okay, let's carry on. We've labeled, and we're going to go into the next slide. Okay, state the number of the region of this plant organ where you find diffusion of gases. We've just looked. Where are we going to find diffusion of gases? We're going to find it here, the region. All right, if you have a look, the region says here, state the number of the region of this plant organ where you find diffusion of gases. Guys, 2 marks. All right, 2 marks. So, where are we going to find it? Here, number 10, and over here. This is where it's going to move in and out, but it needs to go through the stomata. So, number 10 and number 12. See, 2 marks. So, you need to think there, all right? 2 areas. In which numbered part does photosynthesis mainly take place? All right? Have a look over here. Here's just the cell from the side. What is this called over here? Those are the main tissues involved, and we call them palisade. Remember, they're nice and long and skinny. Palisade, mesophyll cells. All right. Now, have a look. How is the part mentioned in question D here structurally adapted for the process of photosynthesis? How is the part mentioned? Okay, this is important. How is it? Now, if you have a look at the mark allocation, if you tell me it's elongated, whoopee, I'm going to give you 1 mark. All right. You need to tell me how. All right. That is the how. All right. And how is it adapted to the process of photosynthesis? So for your 2nd mark, yes, elongated. That's the structure. So what has it got to do with photosynthesis? Increases the surface area. All right. For sunlight. Okay, number, obviously this tells me that there are 3 things. So, whoopee, I've got 2 marks so far. What's the second one? Okay, what was it filled with? Okay, it's filled with chloroplasts. You could say many chloroplasts. I would actually put many in front there. Many chloroplasts. All right, why? Chlorophyll is needed for photosynthesis. Okay, next one. They're parenchyma cells. One of the functions of parenchyma cells is that they are thin-walled. If you have a look, they're not like parenchyma or sclerenchyma or xylem. They've got thin walls. Why would they have thin walls? Oh, yes. Remember? For gaseous exchange. Or we can say for the movement of gases, all right, to be easier. Okay, there we go, photosynthesis. So have a look. What is its function? Photosynthesis. How is it adapted? Are we ready for our last break?
Yes.
Yeah.
Mindsetters, don't forget, I have posted all the information about the Casio competition on our Facebook page. So if you go to facebook.com/learnextra, you'll see that I have all the information about the competition. So if you want to win yourself this awesome calculator, all you need to do is complete the test yourself questions. It's with an entry form on our Facebook page. There's a link to the entry form with all the questions, and you need to fill out all your details as well. You submit that, and you'll stand a chance to win this awesome calculator. So with that said, let's take a very quick break, and I'll see you straight after this. Welcome back, Mindsetters, from that break. Just a reminder, guys, our Learn Extra Help Desk is brought to you by Excel. So thank you so much for bringing us our Learn Extra Help Desk. Guys, please make sure you make use of it. If we don't get through any of your questions during our shows, we've got the Help Desk there for a reason. So make sure you jump onto our Facebook app page, and then you also have the Help Desk at Mindset so that you guys can send all your questions, and then you'll get answered. You see, it works both ways, guys. You must never sweat. We always have your back.
Okay, let's go back to the challenge question. I think it was quite an easy one, so not too much of a challenge this week. I asked you which it says here of a leaf, what would be the substances X and Y? Very important, and it says in that order. So in other words, what moves in to X, what moves out to Y. And if you have a look at the options to you, right, remember a plant very often is opposite to us. So it's going to be number C, because it brings carbon dioxide in, which would be our X, and it takes oxygen out as a end product or waste product. Okay, I hope you guys, wasn't too much of a challenge this week, but I do hope you get it right. Okay, I'm going to turn to this next question. Right, let me just get there. All right. This is quite a nice interpretive question, the question we looked at before. Pretty much straightforward, all right, asking you structure. Now you need to start to just think a little bit. Okay, if we have a look at the question, the graph shows changes in the width. Oh, here we go, pen. In the width, why is my pen not working? There we go. The width of stomatal openings. Okay, so is it big, right, or is it going to be much smaller? in a dicotyledonous plant that is adapted to living in a hot tropical climate. A hot tropical climate. So you need to start thinking, all right, they've given you a little bit of scenario here. Let's see if we can answer the questions. Okay, question number 1. At what time were the stomata most open? Let's have a look. All right, if you have a look on your graph, it says here, here's the time, all right, and here, this is my independent variable, this is my dependent variable. Here's the time that once I can control, all right, but the set, and this was the results. If I have a look, when was it the most open? There's my peak. That's where, right, have a look, just over 2. Now we need to say, but it asks for the time. Please use your ruler here to be accurate. That's 6, that's 7, 8, 9, 10, 11, that's 12. All right, so if we go up here, that's 10, that's 11, and it's in between that. What's in between 10 and 11? All right, 10:30. Oh, my pen's going all wonky here. 10:30. Now, then they've asked you to draw a diagram of the surface view of a stoma to show the guard cells as they would appear at 1 o'clock. Label those features of the guard cell that make them different from the adjacent. See, it's a different slant in the question that we used just now. The question just said to you, how is it different? Here, you need to put that into your diagram. But the first thing you need to do is you need to draw the stomata. You can't just draw any stomata, all right? It says to you, draw it as it will be, okay, at 1 o'clock. Have a look here. There's 1 o'clock. There's 1 o'clock. What do you notice? All right, what do you notice? It is hardly even open. So when you draw it, you need to make sure, okay, that you have, that your stomata, your guard cell, all right, let me do a little bit, let me do the eraser, okay, not so pronounced, here we go. There we go. All right. My stoma. There we go. My stoma. It must have a very small opening. All right. And I know you're thinking, oh, but my teacher said to me, when during midday, when it's very light, all right, there's lots of sunlight, the stoma is going to be open. Look where you are. Hot tropical area. So, number 1, I've drawn it, pretty much what it looks like. All right. This has got a bit of a boob there, so let's fix it. That must go there. Okay, and that go out. There we go, much better. Now it says label those features, not everything, label those features of the guard cell that make them different. Okay, so what am I going to put in? Chloroplasts, because they don't have adjacent, do not have. And when you draw here, you can put slightly, there we go, a thick inner membrane and a thin outer membrane. There we go, and this is a diagram. Always be sure of the stomata at 1300 hours. Always do a heading, all right, or a caption for your diagram. You're never quite sure if they're going to mark it or not. But the most important thing, at 1 o'clock, the stomata were barely open. You cannot draw something, all right, that looks like that. No, the stomata were barely open. Okay, next question. All right, over here. Ooh, as I said to you earlier, we're going to get to this one. Describe how osmosis, all right, influences the stomata. Okay, so during the day, all right, what happens? The chloroplasts make glucose, all right, in turn, which causes, I like to use the word, right, a hypertonic solution. The epidermal cells are hypotonic. So we have here hypertonic, high water potential, right? And here we have a low water potential. Where does water move? From high to low. And my cell membranes, all right, let me extend a bit. My cell membranes are unevenly thick, causing them to part. All right, and because of that, how do we get the opening from one to the other?
And then
Ta-da! Because that's the end of a question.
You're turning into me now.
I'm turning to you. Too much company. Yeah.
Thank you so much, Cheryl, for the lesson. Mindsetters, thank you so much for tuning in. All the instructions to enter the Test Yourself competition, proudly brought to you by Casio, are on our Facebook page. So we'll see you next time, guys. Bye.